This is the second part of the lecture on the basis of the electrical signaling. And now we come to consideration of the action potential. Action potential is the main electrical signal that is used by the nervous system to transfer information between neurons and also to send commands from neurons to the other excitable cells, such as muscle and, in some cases, endocrine cells. For the cells of the nervous system, all their action potentials are normally standard in amplitude and duration, but the sequential action potentials convey various information to and from the central nervous system. You can observe the sending from axon hillock, where action potential is generated, down the axon terminals. So it is an information unit. Action potentials combined with periods of rest and potential maintaining serve to information encoding, similarly to computers encoding, where all the variety of information is encoded by alternating ones and zeros. To be the optimal unit of the information transfer, action potential should meet certain requirements. It must be as short as it is possible and use as little energy as possible. And actually, action potential meets all these requirements, as we will be able to see from the lecture. Action potentials are exactly the fast oscillations of the membrane potential, and each action potential consists of at least two main phases: rapid depolarization, that is followed by the rather quick repolarization, which is restoration of the initial membrane potential. And they actually appear as a spike, and sometimes even the word spike is considered to be the synonym of the action potential. Here you can see the depolarization, very quick, almost vertical increase from minus 60, which is usual resting potential, up to plus 30, which is achieved at the peak of action potential. So this is depolarization, decrease of negativity, which finally is converted to the positive charge. And then, very quickly, there is a restoration, which is repolarization phase, and potential comes back to the initial value. In most cases, there is also the hyperpolarization at the end, but not necessarily. We'll observe in the next slides. Here you can see the action potential recording. Actually, only high spikes are visible here. Also, you can notice that while the action potential frequency progressively increases here from line to line, their amplitude remains practically constant. Voltage-gated channels are the main channels involved in the generation of the action potentials. And let's consider first channels that should be open for the beginning of action potential. And the state of this channel determines the ability to generate action potential. These are fast sodium channels. Fast sodium channels are arranged as the other channels as well as a tube consisting of protein molecule that passes through the whole membrane. But in contrast to many other channels, this channel has two gates. You may observe here on the outer surface of membrane, there is a portion of molecule which is turned in such a way that it blocks the entry to the channel. This is called the activation gate. It's simply part of the protein molecule that is turned in such a way that it closes the passage. And this conformation of this portion of the molecule can be changed so that it can turn and become open. Activation gates. At the inner side of the channel, there is an inactivation gate, which is in resting state, which is open. So the conformation normally is in such a position that it allows passage for ions. But also this conformation can be changed due to changed voltage, membrane potential. Inactivation gate. So it's unusual to have two gates. Only sodium channels have these two gates types. And let's observe. These are selective channels for sodium ions only. Sodium is the main ion of the extracellular fluid, and inside of the cell, concentration of sodium is much less. So when we have the resting state of the membrane, so that inner potential is negative and superficial charge is positive. In this case, the channels maintain their resting state, which is closed. Regardless of open inactivation gates, the passage of ions is not possible because activation gates are closed. But when there is excitation moment, during excitation the charges change, and positivity inside and negativity outside immediately changes the position of both gates, but not with the same rate, and activation gates react faster and they open. When the channels become open, there appears the passage for the sodium, and sodium moves passively down its concentration gradient. So through open channels, sodium readily goes into the cell. This time is very, very short because inactivation gates also react to stimulation, and they very soon become closed. And so, regardless of open activation gates, sodium cannot anymore to pass into the cell from this moment. This channel is considered to be inactivated. Closed state, here it's again closed, but it's different opposite position of gates. So the channel is considered to be inactivated. Let's recall that resting ratio was of permeability for potassium and sodium was 1 to 0.04, and now permeability for sodium exceeds potassium 20 times. And so if we compare to previous 0.04, it makes 500 times higher permeability at the moment of excitation for sodium. And moreover, if you remember, the permeability for sodium also some sources consider as even at rest 100 times high. So if it's 100, it makes many, many times more permeability increase during excitation. So anyway, you can say that there is a dramatic increase of permeability for sodium during excitation. And channels density in the membrane is so high that through many, many channels, a lot of sodium ions are ready, able to come into the cell, regardless of the very short period of open state of channels. Actually, the open state lasts for a few 10,000 portions of a second. For individual channel, this is an extremely short time. So it's very short time gap between activation gates opening and inactivation gates closure. But the number of channels is great, and sodium enters so that potential inside of the cell becomes positive. To generate the action potential, membrane should have both sodium and potassium voltage gated channels. So if certain membrane doesn't have such channels, there is no opportunity to generate action potential. It should be kept in memory for the postsynaptic membranes and some other membranes. Resting state of the channels. For sodium channels, you are already familiar that activation gates are closed at rest, while inactivation gates are initially open. Potassium channels are simpler, they have just one gate mechanism, and at rest this gate is closed. So, and it's very easy to remember that in a resting state, all voltage gated channels, regularly used for action potential generation in neurons, all these channels are closed in the resting state of membrane. But when excitation occurs under stimulation, these channels change position of their gate. But there is a sequence of opening and closure. Successive opening and closure is considered here. Sodium channel initially is closed because of activation gates are closed. But when stimulation comes, sodium channels become open shortly because the activation gates open and sodium is able to pass into the cell. Note that channels always provide a passive movement. Channels are not pumps. And this is the first event that begins the action potential generation, opening of channels. Then very soon sodium channels become inactivated. So It's a closure of inactivation gates. And just from this moment, potassium channels become open. Initially, they are closed in the resting state, and now this is the event number 3, opening of potassium channels. So if you compare the gate mechanism, rate of opening or closure, the fastest gates to react to stimulation are sodium channels, activation gates. They open first. Then there is the reaction of the inactivation gates to the same stimulation. It's just a little bit slower, and as a result, sodium channels become closed, and between these two successive events, there is a short time when sodium channels are open, so that sodium influx into the cell occurs. Then the third event, this is change of position of potassium channel gates, which become open. And potassium again starts to move down its gradient from the cells outside. Therefore, positive charge, which was achieved after sodium entry, now can be lost. So it's all occurs passively, and it's much faster to restore the initial negativity by release of potassium out from the cell down its gradient. Because the other way to move out sodium would require much longer time and much more, and it requires energy. Of course, this process occurs normally due to action of the pump, but we can do it later, and potassium produces repolarization quickly, and it's more important to be repolarized so that opportunity and probability of new generation appears again, new action potential generation. And we should not wait until the pump finishes its work. We can generate impulse after impulse, regardless of pump operation at the moment. So, let's see sodium and potassium conductance during the action potential. First to react are sodium channels. Here the conductance is shown as a result of numerous channels opening, which open not perfectly simultaneously, with slight difference in time, slight difference. Therefore, it's wider than action potential. Then single action potential comes up and down sharper. So sodium conductance is first, and exactly while sodium channels are high amount of open channels we have, because conductance directly depends on number of open channels. And you see during this sodium conductance, action potential first phase occurs. And after the peak is reached and sodium channels start to close, exactly by this time the potassium conductance starts to increase. So potassium conductance comes later, and its increase is later. And so, due to sodium conductance, we have the first phase of potential, which is depolarization, and then second part of it's due to potassium going out of the cell. And also you can notice that potassium conductance stays a longer time open. They are slower opening channels, and correspondingly they close slower. And therefore, at the moment when resting potential is achieved again, these potassium channels are still open, and potassium can go more than necessary to restore potential, and it creates one more phase, which is hyperpolarization. Why potassium continuously leaves the cell, even at the level of normal resting potential? Because potassium difference exists, and the more permeability is for potassium, the more potassium will leave the cell, and it's higher than normal resting permeability for potassium, because many channels are closed still, and many channels are open still, some are closed. But so potassium continuously leaves the cell and produces excessive negativity in the cell temporarily. But when all the channels, voltage-gated potassium channels, become closed, everything becomes restored back to normal resting level. So you can observe here the 3 phases. And actually the second and third phases, repolarization and hyperpolarization, are determined by one and the same process, potassium loss from the cell, and it all goes due to longer time open state of potassium channels. Therefore, we have the three phases of action potentials. In some cells last phase is absent, but mostly in the neurons there is third phase. So first is depolarization, when negativity decreases, and actually it's not only decreases, but comes to the opposite charge, which is reversed potential, and so it comes to the positivity, depolarization. Then repolarization phase restores the potential from positive charge back to normal resting level. But at this moment, potassium channels stay open, and due to this, additional loss of potassium from the cell results in hyperpolarization. So total amount of phases usually comes to 3 phases. When we'll study the action potential of the skeletal muscle cells, you will see that there is no hyperpolarization. But for neurons, usually there are 3 phases. Let's draw the figure of the action potential. First, it's necessary to put axis, and the vertical axis will show potential. And resting level is minus 60 millivolt. Actually, you remember, from minus 60 to minus 70, it can be dependent on the neuron. Then 0 and peak will amount to plus 30 millivolt. Then time goes along the horizontal axis, and also it's necessary to remember one more thing, that the so-called threshold level. We'll talk about this. But first, let's observe the sequence of events. First thing, inside the cell is charged negatively and positively at the surface. And first phase of the action potential is sodium entry through open channels. And as a result, sodium brings charge from minus 60 to positive plus 30. And this is the end of the depolarization phase, and this is the potential reached inside the cell at the peak of the action potential, plus 30. So this is the first phase, depolarization. Now, by the time when sodium channels become inactivated, potassium channels begin their opening, and through open potassium channels, potassium goes down its concentration gradient outside of the cell. And it quickly brings the charge to resting state, and it doesn't stop at this moment, as I do for demonstration, but continuously goes out. So due to this first restoration of the initial resting potential, from the peak down to resting potential, this is phase number 2, repolarization. But potassium permeability stays high, and potassium continuously goes from cell. And in this case, it creates a third phase, which is hyperpolarization. Let's turn back to the threshold. So again, sodium enters the cell. This is first phase. Then potassium leaves the cell. So you see, both events occur passively. It's enough just to open channels and to close channels. Of course, after each action potential, there is little deviation of initial difference of concentrations. But really, it's such a negligible difference after single potential that even hundreds or thousands of potentials do not disturb greatly the difference that exists normally between intracellular fluid and extracellular fluid. So we are able to generate numerous potentials, generate them with high frequency, and still the pump operation should not be so quick that to restore immediately everything as it was before. Generally, pump doesn't work within the first phase, depolarization, and after the peak, the pump starts to work. But we don't depend on the pump work directly, because we can generate new impulses after each potential. So, and all is repeated and repeated. And pump should work because anyway, after certain time, there will be serious change of the initial concentration gradients. So pump is necessary, its work is necessary. But we don't depend directly on each individual moment on the pump work, fortunately. Then, threshold. Notice that it's about minus 45. It can be between minus 40, minus 50, depending on cell. And this is important level which must be reached in order to generate action potential. If stimulation is weaker and this threshold is not reached, action potential does not develop. Let's observe the situation when we use minimal necessary force. It's called the threshold stimulus. In this case, the generation of action potential is not as quick as it is shown at the initial figure. And in this case, the depolarization slowly starts to decline, but finally at the upper point, it reaches the threshold level. And this is minimal force, which is enough to generate action potential. And when this point is reached, then sodium channels become all open. Generally, the threshold level is considered to be such a charge of the membrane, such a potential, which results in guaranteed opening of all sodium channels present in the membrane. And when this moment is reached, immediately generation of action potential occurs. So it just moves slightly later with smaller force. It produces action potential slightly later. But if we choose any smaller than threshold stimulus, then depolarization will go and it doesn't reach the threshold level. And it remains just a very short time depolarization without resulting action potential generation. Let's observe the threshold of depolarization. What is the basis for the threshold? Why exactly it develops at certain level? This certain level, like minus 40, minus 50, was empirically found. But what underlies the threshold? You see, the number of open sodium channels should be enough for the closed circle formation. Depolarization, which is initial stimulus, opens certain number of sodium channels. And this first depolarization through opening of sodium channels contributes to further depolarization, because through open channels, sodium enters the cell and sodium brings depolarization because it's charged positively. So if this influx creates depolarization stronger than initial one, so it contributes to further depolarization, and almost immediately all the channels become open. All the channels. But if initial stimulus causes depolarization, which opens not enough sodium channels, so that entry of sodium will produce depolarization less than stimulation depolarization, less than initial. Because initial depolarization will not last forever. It lasts for a short time. It manages to open some sodium channels, and this depolarization disappears itself. Just through open channels, sodium comes into the cell and creates depolarization. And when it exceeds the initial one, then all the rest channels will be immediately open, and this is considered to be the level of the threshold. So, and this level is about minus 45 plus minus a little bit, from 40 minus to minus 50. And why we care about threshold? Because excitability, ability of membrane to generate action potential, depends on the threshold. In its resting state, excitability exactly is estimated by the level of the threshold. The lower the threshold is, the easier to produce action potential, so the higher excitability is correspondingly. It's the usual way. Thresholds are always the lower, the better. So it corresponds to high excitability, to higher something else, depending on which threshold we consider. Look at this one. For example, we have resting potential minus 60 and the threshold minus 45. Before we proceed, you should be aware that threshold is a word that sometimes we mean by threshold exactly the absolute level of depolarization, critical level, like minus 45. This is absolute value of the critical for depolarization level. But in many cases, under the word of threshold, we understand the difference, this difference. So it's two types of understanding of the word of threshold. And mostly we estimate threshold as a difference. And now, for this example, a difference makes 15 millivolt. So if we depolarize the membrane by 15 millivolt, the action potential develops. If we depolarize some more than 15, anyway, action potential develops, and usual size it will have. But if depolarization does not reach this 15 millivolt, so any value less than 15, there will be no action potential at all. This depolarization will disappear without any result. And let's see, another case, the other cell has resting potential minus 70. And the threshold level, let's admit it is the same, minus 45. Then threshold is higher, and now it makes 25 millivolt. And then, to stimulate such a cell to reach the action potential generation, we should choose the stronger stimulus, at least 25 millivolt, no less. So the stronger stimulation is necessary, the less is excitability. So higher threshold corresponds to lower excitability. So here excitability is less as compared to previous situation. Then, in a resting state, we can estimate easily the excitability by the threshold. But excitability depends also on the number of sodium channels ready to open. Just in resting state, we assume that all channels are ready to open. But this is resting state only. For state of excitation, it is different. During action potential generation, excitability changes greatly because the state of sodium channels is changed. And if we observe these situations, two cases, activated channel or open and inactivated channel. Both open and inactivated channel, they both cannot open again before they restore the initial position of gates. Only the state of channel that is shown to the left, only in this state the channel can be opened. When activation gate is closed and inactivation gate is open. So open channel to respond to new stimulation should first be closed and then restore position of gates. The inactivated channel should again restore the initial position of gates, which means that first activation gate should close, then inactivation gate should open. Only after this channel may respond to stimulation by opening. So temporarily, exactly in the open state, as well as in inactivated state, the channels cannot respond to new stimulation. At the moment, they are busy with response to previous stimulation, but new stimulation will pass unnoticed by these channels, so there is no any excitability at this moment. Excitability is zero for both situations, open activated channel and inactivated. And this period of absence of response is called refractory period. Actually, the word itself means no response, no excitability. And during excitation, we have such a period because the channels are busy with response to previous stimulation. So new stimulation, they cannot even notice exactly within this short moment when channel is open or channel is inactivated. So when membrane is excited, for a short time, there is no excitability. It becomes simply zero. Then excitability is quickly restored to normal level. And for a very short time, it becomes even greater than normal. So all this depends on the state of the sodium channels, the opening of which is a response to stimulation, the previous one. And new response is again opening of these channels. So open channels are not able to open as well as inactivated. So first situation, open channels as it occurs during depolarization phase, and inactivated state of the channel, which corresponds to the peak of the action potential. So let's recall again all the channels states during generation of action potential. Initially, excitability is normal. At the moment of resting potential, all channels are able to be open and the threshold is normal. Then, so this is a simple initial closed state of the channel, ready to open after stimulation. And first phase which develops due to open state of sodium channels. Potential goes upward to positive values exactly because sodium goes through these channels, open channels, into the cell. And open state of channels means full absence of excitability. It's type of refractory period which is called absolute. Absolute refractory period means that excitability is not low, but it's absent at all, zero excitability. Similarly, at the peak, inactivated channels also are unable to respond. So from the point of threshold until the point of peak, including this moment, there is no excitability at all. It's absolute absence of response or absolute refractory period. Then, second phase, repolarization. During repolarization, we have the process of reactivation of channels. For their reactivation, they should successfully pass into the state of totally closed gates, and only then reactivation process results in restoration, because it's necessary finally to open the inactivation gates in the inner side of the membrane. When all channels are restored, it corresponds to full restoration, but they do not do it perfectly simultaneously. Therefore, there is a period when, for example, 50% of channels restored, excitability is 50%. 70% restored, excitability is 70%. So during the initial part of repolarization, when channels are reactivated, excitability increases from zero level to normal. And so this is decreased excitability until the moment when we reach full excitability. And here I omit a little portion of the last repolarization part when excitability exceeds normal, but we'll come to this in the next slide. So hyperpolarization decreases excitability. Channels are restored, but the threshold now is higher than normal. Compare, this is normal and now it's high. It's a simple hyperpolarization, which makes the potential to be far from the threshold. So this is accompanied by decreased excitability. So now again, relation of action potential and phases of excitability. Depolarization corresponds fully to the absolute refractory period. Excitability is exactly zero. Repolarization, main part of repolarization, corresponds to the relative refractory period. So it's not zero excitability. It is now more than zero, but still less than normal. And it quickly increases, but before it reaches normal level, it is a relative refractory period. Then, in the very end of the repolarization phase, when the potential passes nearby the threshold and comes before threshold, between threshold and resting potential, excitability for a short moment becomes higher than normal. So it's called a supernormal excitability, slightly higher than normal. So it becomes even easier than usually to excite the cell, to excite the membrane. And hyperpolarization corresponds to lower, decreased excitability, and it's called subnormal excitability. Supernormal before and now subnormal. Excitability becomes less proportionally to the degree of hyperpolarization. Usually this is not very deep hyperpolarization, so excitability decreases not so much. And now again, phases of excitability shown in the figure. As you can see, just in parallel to the action potential, here beneath you can observe the figure of excitability. This is an action potential with its phases and the normal values for resting potential, threshold, and peak are shown here. And beneath we have line which corresponds to normal excitability level, and here normal excitability is taken as 100%, but the whole range from 0 begins up to 100% for excitability. So first phase of action potential is depolarization phase. And basically we start to consider this depolarization of action potential from the moment of threshold, because before threshold it's not fully clear will be potential or not. And so before reaching the threshold, when potential becomes closer and closer to the threshold, approaching, simultaneously excitability increases because there is higher and higher probability of generation of action potential. But phases of excitability we start to consider from the moment that corresponds to the point of threshold for action potential. Correspondingly, we start from the first absolute refractory period, this line that corresponds to exactly 0. So from the moment when the action potential reaches the threshold, minus 45 millivolt in this case, the excitability immediately drops down to 0, from even higher than normal. At this moment, when all channels become open, it goes to the opposite direction. Excitation is in progress, but excitability becomes 0 because this excitation is a response to previous stimulation. And if we try to stimulate membrane at the very moment, there will be no changes. No stimulation or very strong stimulation, kilovolt, doesn't matter. The membrane is busy, it produces response to previous stimulation. And new stimulation will come unnoticed. So because of absence of excitability temporarily during this action potential generation, during an absolute refractory period, as you can see, is rather short. It corresponds to the peak duration, and usually it doesn't exceed 1 millisecond. It's a very short period of absence of excitability. Then from the peak we begin the initial portion of the repolarization, and during this moment there is a restoration of initial gate's position, and the 4 channels are in the process of reactivation, and it results in the relative refractory period, which is restoration from 0 to norm. And correspondingly, during this phase, excitability is less than normal, but not 0. So we call it relative refractory period. If you compare this very short period beginning, it's much harder to excite the membrane, but theoretically possible. Closer to the end of this period, it will be a little bit more difficult than usually, but still more possible. Because, for example, at the moment of 80% of excitability, it's rather easy to excite, but it's necessary to use stimulus stronger than normal, a little stronger. Then, last portion of the repolarization 2B part corresponds to the moment when we pass nearby the threshold, and here a very weak excitation, very weak stimulation can result in generation of action potential, because by this moment all the channels are already restored. They are restored very quickly. And the potential is close to the threshold, so little stimulation can be enough, smaller than normal, minimal, can be enough to produce new action potential. So new action potential, you see, may start much earlier than the end of this hyperpolarization. It may start from the beginning of the 2B phase. So action potentials can be like serrate figure. Up, down. Maximal rate is limited practically by the absolute refractory period. This is the most important. The duration of the refractory period determines the maximal rate of impulses. And as this period is extremely short, maximal rate is extremely high, therefore. So, but let's finish. This is supernormal period of excitability, when we say excitability exceeds the normal initial level. And it ends with the cross-section of the resting potential, where the hypopolarization starts. And hypopolarization pulls the level of potential far and far from the threshold. Therefore, excitability goes in parallel downward. It simply goes in parallel and comes to the normal level in the end of hyperpolarization. And this is subnormal excitability. Now, what is the conclusion from these phases? What for we check it? Because during absolute refractory period, there is no any opportunity to generate action potential. No stimulus can produce the action potential because it's absolute absence of excitability. And exactly this is the limiting period. Maybe plus little beginning part of the relative refractory period also can be included. In some simplified classification, they combine both absolute and relative refractory periods into one refractory period, and consider this is the main limit for maximal rate of impulses. Then relative refractory period is now different because stimulus much stronger than normal is able to activate the membrane and produce action potential. Less channels are ready to open, but with very strong stimulus, finally we can reach the threshold and produce action potential. Then, supernormal excitability. This period is very short, but nevertheless, during this period, we can use to produce action potential stimulus even weaker than normal. If normally we establish the threshold force necessary to reach the threshold. And now, stimulus which is less in force than threshold. Some may be enough for generate action potential. And finally, subnormal excitability. Subnormal is less than normal, so stimulus should be a little stronger than normal, than usual minimal threshold stimulus to produce an action potential. All these changes are finally come to the normal excitability level, and usual threshold stimulus is enough to generate new action potential. So, but within the action potential, the excitability changes dramatically to zero. It drops exactly vertically to zero in the beginning of action potential. Then very short time it stays zero, and then it quickly restores and makes little variability around the normal level. And then it comes to normal initial. But notice that we can generate action potentials one after another without waiting until hyperpolarization is over. Just in the end of relative refractory period, the new potentials can be generated. Not to say about supernormal excitability, which is the perfect for excitation. And here we can see that the action potential follows the excitability law, which is called all or none. Many potentials follow the opposite law of force, where the stronger stimulus is, the higher amplitude of potential is achieved, but this refers to other potentials, not to action potential. As you could see before, here we progressively increase the force of stimulation, and this length of these green lines shows that force becomes higher and higher and higher. And these first few are subthreshold stimuli, and as you can see above, these potentials do not reach the threshold. Therefore, action potential does not develop. So none, no action potentials at all. All or none, sometimes it's called all or nothing. So nothing develops, as we can see no action potentials. Let's make the next stimulus threshold. It's the minimal necessary for reaching the threshold level. And you see action potential develops. Here it looks like loop-lap, and it comes up vertically, but it's simplified picture. So, and then when we increase stimulation stronger and stronger, nothing changes in amplitude. There are no changes in amplitude, and standard size of action potential can be observed. This is all. And so there can be half of potential, 70% of potential. Potential is present or it's absent. All or none. So difference, subthreshold stimuli cannot produce any response, none. Threshold stimulus, as well as any higher, stronger stimuli, they all produce one and the same full action potential. It's full amplitude to plus 30. So all or none. This is main law, or when we say all or nothing, main law that action potential follows. So, on the basis of all that we considered before now, action potential, as you can see, meets the main requirements for the optimal information unit. Because of very fast opening and closure of channels, the potential itself takes very short period of time. It may last for a few milliseconds, but its minimal duration is limited by the absolute refractory period, and it's about 1 millisecond or so. So it's a very short time taken by single unit of information. Then, channels provide the passive movement of ions, and therefore we don't spend much energy for generation of the potential. Of course, we cannot forget about the pumps, because pumps should work constantly to keep the concentration differences, which finally make the ions to move passively through open channels. And energy, which is spent for pumps working, actually it's a great amount of energy. General estimation gives values about 40% of all the energy we spend in the body, and this energy, 40%, is spent for pumps work, because pumps should move ions against their concentration differences with usage of energy by ATP splitting. So, but it's pump working, but exactly per one action potential, we do not spend much energy because basic movements of sodium and then potassium is produced passively. So it doesn't take much energy for one potential, and even for thousands of potentials, we don't use much energy. Therefore, action potential is very good unit of information. It's energy saving and time saving, and it enables us to react quickly and easily to different changes in the surrounding environment. It enables us to spend not much energy for generation of many impulses, which can be basis for thinking, for decision making, for control of skeletal muscles, and for many other processes where action potentials are involved. So this is energy saving and time saving unit of the information transfer by the way of the electrical signaling in the central nervous system. And again, action potentials are the nervous impulses. In general, we can say simply that these are nervous impulses that are sent from neuron to the other neurons, from receptor neurons to neurons in the central nervous system, and then from neurons to the other excitable cells, mostly to muscles, but also some endocrine cells are under control of nervous system, such as, for example, catecholamine producing medulla of adrenals releases adrenaline, noradrenaline in response to sympathetic nerve stimulation. All this is the electrical signaling, which is faster and very precise. Again, neurons generate impulses in the area of the axon helix, in the beginning of axons, and then impulses are conducted along the axons and finally conducted through the synapses. But these processes are entirely different story, which can be considered in the next lectures, how the impulses are conducted down the axons of different types, and how these impulses finally reach the target cells through the synapses, specific structures that conduct impulses from nerve axon terminals to the next target cells. So, but this is a way to transfer information very quickly, up to 120 meters per second. Conduction velocity can reach. And this is the end of the basics of electrical signaling. Thank you for your attention.